Hello friends, welcome back to my channel Xgene and Proteinx. In this tutorial video, I'll discuss about the overview of the immune memory against SARS-CoV-2. So before going to the main discussion, I would like to discuss something about the basic concept about the immune response. So the immune response is arise when any pathogens or bacteria or virus when infect the cells. Afterwards, the immune cells it induced or activated against that pathogen, and it destroy that pathogen. So eventually, that the infection would be cleared. So this is the immune response, and this is the naturally occurring. So I gonna show you that how many kinds of immune cells are there. So there are different kinds of the immune cells, but there are some basic and very important three different kind of immune cells are there. These are CD4 positive T cells. This is also known as the T helper cells. So these kind of cells are important to produce the cytokines, mainly the IFN gamma, and also it activates the T cells as well as it stimulates the B cell to produce the antibody. And another kind of the T cell are the CD8 positive T cells. CD8 positive T cells. These cells are known as also cytotoxic T cells. So these kind of cells it destroy the infected cells upon its activation, and it has some several mechanism. Like the main mechanism is the perforin granzyme. So the CD8 positive T cells, they produce this kind of perforin granzyme, and these are the cytotoxic materials, and these kind of materials they destroy the infected cells. And another kind of cells are the B cells. So B cells are normally they produce the antibody. So when the B cells are activated by the CD4 positive T cells, and then B cells it produce some kind of specific cells that are known as the plasma cells, and plasma cells they produce specific kind of antibody that very much precise and selective against the specific kind of pathogens. And this kind of antibody, they destroy the pathogens. So this is how it works, the B cells. All right, plasma cells. So here is the overview of the activation of this kind of cells. So here, as you see in this figure, these are the CD4 T cells, T helper cells. So here also this kind of cells it activate B cells, as I mentioned here in this slide, that there are some activation, some process like CD40 and OX40L. So this kind of receptors are there in the B cells, and when it bind with the ligand, then B cells get activated, and when it get activated, then it gonna produce some kind of specific antibody against that specific antigens. Because you see here the virus are here, so they're gonna recognize with their B cell receptor, and this kind of pathogens are very much important for inducing this kind of antibody. And when the B cell when they produce the antibody, these are very much selective against this kind of specific virus, and thereby there are a lot of antibody, and this kind of antibody producing cells are known as the plasma cells. So when any selective kind of cells they produce antibody, then these selective kind of cells they replicate and they proliferate, but the rest of the cells they destroys. All right, then this kind of plasma cells they survive, and then it produce this kind of specific antibody. And in another case here you see the CD8 positive T cells. When it get activated by the by the antigen presenting cell. You see here, this is the dendritic cell or antigen presenting cells, and antigen presenting cell, it has the MHC complex. So MHC complex, it represent the antigen, and this antigen, it gonna it bind with this kind of cells, T cells, and when this kind of antigens are recognized by this kind of CD8 T cells, and also there are some other ligand as like B cells, they also have some other kind of TCR signals, CD28, CD27, CD40, these kind of signals, and when these kind of receptors they are bind with the ligand, then CD8 positive T cells, they get activated, and when they get activated, then the proliferation of the CD8 positive T cells occurs, proliferation and the activation, and then these CD8 positive T cells, they produce perforin and granzyme, and by using this kind of cytotoxic materials, they destroy the specific kind of pathogen or infected cells. So this is how it works. And also here you see the T helper cell, it gonna produce some kind of cytokines like IL-2 and IFN gamma. So by this kind of cytokines, also CD8 positive T cells get activated. So there are different kind of some process, and this is the several series of mechanisms. This is how CD8 positive T cells get activated. So this is about the basic concept about the immune system and immune response. So let's move on to the generation of the memory T cells. So how this kind of memory T cells, it occurs actually, how it generated. So first I have to start from the naive T cells. So you know, these naive T cells are the T cells that not previously respond to any kind of pathogens. So these kind of T cells are known as the naive T cells. But when these kind of naive T cells, they're gonna start responding against any specific kind of pathogen, then they produce some kind of effector T cells. So these effector T cells are the cells that gonna respond against any specific kind of pathogens. And when this kind of T cells, they proliferate against this kind of pathogen, then these pathogens or infected cells, it destroyed by this kind of effector T cells. However, there is another important point I would like to mention here, that over time, when this kind of virus or pathogens are cleared from the human body, so beside this, also this kind of effector T cells, they're gonna decline, and the number of the T cells, number of this kind of effector T cells, it reduced over time. But the important thing is that, and very surprisingly, the very small population of the T cells, they're gonna survive and they're gonna develop as the memory T cells. So this kind of specific kind of T cells, these are very much trained, and these T cells, they proliferate when this kind of reinfection occurs. So before this kind of reinfection, this kind of T cells are not gonna proliferate or not gonna produce an effector T cells. Alright. So here you see the naive T cells first, and then the cell division occurs, and then after cell division, the effector T cells occur. Effector T cells is generated. So these effector T cells are for it induce the immune response. Alright, it induce the immune response. Immune response. So this kind of effector T cells, it induced immune response. But after several days, when this kind of pathogens are completely cleared from the human body, then this kind of effector T cells, they're gonna decline, or this kind of T cells, they die. But there are a small portion of the T cells, they they survive for longer time period, and this kind of T cells are known as the memory T cells. So this kind of T cells, they're gonna produce effector T cells, or this kind of memory T cells, they're gonna stimulate the B cells until the reinfections occurs. Alright, so before reinfection, this kind of memory T cells, they does not respond to any kind of pathogens. So this kind of trained T cells, trained memory T cells, their job are very much specific. So when this kind of memory T cells, when they are start responding only when reinfection occurs. So this is the main difference between the memory T cells and the normal effector T cells. So now I'm gonna show you that the different kind of the memory T cells, like the CD4 positive T cells that I already mentioned, their job also for as a T helper cells. They induce or they produce cytokines, and they activate T cells, cytotoxic T cells, and the B cells to produce antibody. So from this kind of CD4 positive T cells, the small portion of the CD4 T cells, they survive for longer time period, and they formed as a memory CD4 T cells. These cells also very much trained, and over time, when the reinfection occurs, then this memory CD4 T cells, they recognize against this specific pathogen, and they stimulate the B cells to produce antibody. So these B cells, they produce plasma cells, and these plasma cells are very much specific, that can be produced, that can be generated the antibody. So this is the memory CD4 T cells, and also the similar kind of cells are memory CD8 positive T cells. So memory CD8 positive T cells, these are also the similar kind of their job, like the CD8 positive, the effector CD8 positive T cells, the small portion of these cells, they survive, and when the pathogen they infect, then these memory T cells, they proliferate and they destroy this kind of pathogens. So this is how it works. So this kind of memory T cells, they actually remember their past event, and the pathogens they can recognize. All right, and also the memory B cells. This is the memory B cells. This is the memory B cells. So memory B cells also, this is like from the B cells, some small portion of the cells are survive for longer time, and they can be induced, or they can be activated when this kind of similar kind of pathogen they reinfect. So when at the time of this upon of this kind of reinfection, these memory B cells, they get activated and they multiply, they proliferate, and they produce this kind of specific kind of antibody that can be destroy that specific kind of pathogen, because this memory B cells also trained. So these all kind of memory cells are very much trained and very much specific to destroy that specific kind of pathogens. They also can destroy the specific kind of pathogens, as well as they can also produce antibody, specific kind of antibody that can destroy that kind of specific or selective pathogens. All right, so this is, their jobs are very much specific. So now I'm gonna show you that how the immune memory is formed against SARS-CoV-2. So as I mentioned earlier that how this kind of memory T cells or memory cells are formed. So here also the same kind of things. So when the primary infection occurs, like SARS-CoV-2 when infect any host cells. So here you see this is the virus SARS-CoV-2. This is the SARS-CoV-2. So when this SARS-CoV-2 it infect any host cells, then upon their infection, there are a lot of immune cells they get activated. So you see here all of the T cells, B cells, antibody producing cells, these all are get activated and they proliferate and they multiply. So when they multiply, then they destroy this kind of patho SARS-CoV-2 virus. Alright, but over time, after several days, when this kind of virus are completely removed, completely eliminated from the human body, so at that time the number of these T cells or B cells, their numbers are gradually decrease. They're gonna die over time period after several days when this kind of SARS-CoV-2 they get eliminated from the body. So but from there, from this kind of cells, also the small percentage of the cells they produce, they can survive for longer time period, and this kind of cells are formed memory T cells. So memory T cells also formed here, and also as well as memory B cells also formed here, right? So now what if the reinfection occurs? So when the reinfection occurs, then in the human body they already have this kind of specific memory T cells and specific B cells that can recognize this specific kind of SARS-CoV-2 virus. So when the similar kind of virus they reinfect the second time, then this in the host cell, these memory B cells and memory T cells, they can recognize this kind of pathogen, this kind of virus. They can detect it. So they can detect it. So they can detect, and afterwards they multiply. So when they multiply, then their job it gets started. So what their job? Then their job is the memory CD4 T cells. What they do? They do cytokine release like IFN gamma, IL-2, this kind of cytokine they release, and they also stimulate B cells, and B cells gonna produce some specific kind of plasma cells, and then plasma cells they're gonna synthesize the specific kind of or selective kind of antibody. As well as also the memory CD8 positive T cells, what they do? They also produce some selective very cytokines like perforin and granzyme B that I mentioned in my earlier slide, and by this kind of cytokines they destroy the virus, right? Because these all are trained and very much specific. As well as also the memory B cells that forms these memory B cells, they know how to produce this kind of antibody, this kind of selective antibody. So they produce some specific kind of plasma cells, and these plasma cells also produce SARS-CoV-2 antibody. Alright, so this is how it works. So now I'm gonna show you some recent study that people they executed. So here you see this paper already published, and this is published in the Lancet. So here what they did actually, the COVID-19 patient from the different kind of patients, they divided into different kind of the category, and they collected the samples. So there are samples containing like 9 months and 13 months of the infection of this COVID-19 patient sample, as well as also some other kind of samples. So let's move on to the next slide. I'm gonna show you the result of this paper. So here, as you see in this figure, that the HV So this is the normal individual, no infection. So here you see that the HB. So this is the normal individual, like no infection is observed in this kind of volunteer. And the vaccine, so this is kind of vaccinated individuals, and the infected individual, like the people who wanna infected and not longer period of time. This is the shorter period of time, and this is the M9 and M13. These are the after 9 months of the infection, this kind of individual and the individual who got infected. So after 13 months they collected the samples. So there are 1 2 3 4 5 different kinds of the samples. So after their experiment, what they observed that the proliferating T cells, the against nucleocapsid, against membrane, and against spike. So what they found that the 9 months and 13 months later of the infections, the sample that they collected, in that sample the proliferating T cells are very much high compared to the other samples. You see here, this is very much significant data that in all cases, like you see here 9 months and 13 months. So here the proliferating T cells are very much high compared to the infected individual, like shorter duration of the time that people infected. And also here this is similar kind of sub results, like in the after 9 months and after 13 months, this individual samples contain the proliferating T cells are very much higher. So what does it means? So it indicates that memory T cells formed, and these memory T cells, it detected after 9 months or 13 months. But in case of the shorter period of time, the individual that got infected, in their sample the proliferating T cells is lower compared to this kind of longer time period of time. And as well as they also showed that IgG antibody, also the IgG antibody in case of the longer time period, in 13 months, after 13 months, that samples it contain it's higher, the highest, it's very much significant. But if like they did here only the 9 months and 13 months, but you see here in case of the 13 months, this is the most significant amount of the antibody they detected. So it means that the plasma cells or memory T cells or memory B cells, it's higher in this case in 13 months these samples. All right, so this is very much surprising. So I want to show you the another data. So this is the another kind of experiment they did, like exposure to SARS-CoV-2 generate T cell memory in the absence of detectable viral infections. So this article it published in the Nature Communication. So they also found similar kind of results, that in the T cells or memory T cells is developed in case of the higher longer duration of the time period. So, and this is the another kind of paper in the literature I found this, and here you see here this is the different kind of data. When the primary infection occurs, this is a virus. When the primary infection occurs, then the T cells it developed, all right, and these T cells gonna it eliminate this virus from the human body. So over time period, you see this graph. So over time period, when there is no virus, then the T cells population is decreased, but it contains some memory T cells, all right. So when in the time after several times, when the reinfection occurs, then these memory T cells, they're gonna proliferate. So here you see the proliferation. So there are higher number of these T cells, and these T cells, they recognize this specific virus and they destroy this virus. And the number, this is the three different scenario. So this is the scenario 1 and this is the scenario 2. So in this case, what is happening here, you see, in case of the secondary infection, when the T cells population are higher, so in this case, if the but there is one important point I would like to mention here, that in some cases the immune response are not that much sufficient to eliminate or destroy the pathogens. So in this case, the same things happens here. The T cells, they cannot destroy this virus. So what does happen then? This is the chronic infection occurs in this case. So the memory T cells, they cannot eliminate or cannot destroy the pathogens. Alright, then the viral population will be higher, viral load will be higher, and it might be the chronic infection occurs here. And scenario 3, what happening here? So what is happening here, when the primary infection is very much crucial, is very much stronger, so in this case the immune cells, they're gonna proliferate rapidly, and the memory T cells, number of the memory T cells is very high. So this is very much dangerous. Actually, it may arise the autoimmunity. So this is very much dangerous situation in this case. So there are three different scenarios I discussed here. So this is all about this immune response and immune memory against SARS-CoV-2 virus. So if you like this video, kindly hit the like button, share it, and subscribe my channel. Kindly, if you find this video is helpful, kindly subscribe my channel, because your subscription, it motivates me to produce more interesting videos. Thanks.